So let's talk about the urinary system. Just so you know, a reminder, if you look closely at the third lab packet, it does include urinary gross anatomy as well as the histology of everything. There's not a lot on the third practical about it, but there is some. So not a bad idea to at least watch this video before you try that. So the urinary system is very important because it's an important waste removal system, and it's also considerably interconnected to other homeostatic mechanisms. And what that means is that when there is dysregulation of the urinary system, other stuff tends to go wrong pretty quickly. So we'll touch upon some of those pathologies as we make our way through the urinary system. But in overview, we've got two kidneys. The left one's usually a little bit higher than the right, and that's typically because the right side has the right lobe of the liver that hangs down, and it kind of pushes everything on this side down. There is one ureter per kidney, and these ureters are going to take urine to the urinary bladder, which stores urine until it can be expelled via the urethra. So functions of the urinary system are many, as you have no doubt noticed by now, having made your way through 241 and 242. No body system does just one thing. They all do a lot of things, which makes management of them and understanding of them challenging. So that's your challenge as an A&P student, and that's clinicians' challenge to figure out what goes wrong when something does go wrong. So obvious function of the urinary system is in its name, produce urine. But why do that? Well, you have wastes, especially ones that contain nitrogen, and those wastes need to get out of your body and away from your cells and tissues. And that's because nitrogenous wastes are toxic in excess to your cells and tissues. So you don't want too many of them. You also need to be able to regulate blood volume and blood pressure. So blood volume means how much blood do you have in your blood tubes, and that has an impact on pressure because, of course, pressure is just how hard your blood is pushing on your tubes. By selectively excreting ions into the urine or holding ions back, the urinary system can regulate the concentration of ions in your blood plasma, which, as you know, is very important. So we talked about this in this class as early as Chapter 6, when we talked about bone tissue and the regulation of calcium. So that's kind of a big idea there. Helps to regulate plasma pH, along with the respiratory system, as you know, and conservation of nutrients. So you have nutrients being transported in your blood. You need to filter your blood using your kidneys, but you definitely don't want to pee out those nutrients. So you did all this work to, for example, break down proteins into amino acids. You don't want to pee those out. That wouldn't make any sense. So one of the features of the kidney that is unique, or at least remarkable, is the blood and nerve supply. So the kidneys receive a disproportionately large percentage of cardiac output. So between 20 and 25% of cardiac output. So let's take a moment to appreciate this number. That means 1.2 milliliters per minute, or 1.2 liters per minute, excuse me, 1,200 milliliters. Whatever work your heart is doing, 25% of that, a fourth about, is going to your kidneys. So they're extremely vascular, as you can see over here. And that's because, of course, their whole job is to filter stuff out of your blood and then take some stuff back. So they're vascular for two reasons. One is their job is to filter blood, so blood needs to go there, needs to leave, but also because, as you might imagine, filtration and then fine-tuning of what's in the urine costs a lot of ATP. So they need cardiac output for that reason as well. So the kidneys receive and drain a lot of blood. They're also vulnerable. So your kidneys are retroperitoneal, meaning they sit on the posterior body wall outside of the peritoneal cavity. And although they're high enough up that they're guarded a little bit by your lower ribs, they're not completely protected by your thorax. So they're vulnerable to impact trauma because they're superficial compared with other organs. So for example, impact injuries from falls or from car accidents, one thing that first responders and emergency personnel need to pay attention to in those folks is watching for kidney damage and watching for spleen damage, because lacerations, which means splitting by impact or blunt force, to those particular organs can cause the patient to bleed out really quickly. And we don't want that to happen, of course. So the nerve supply to the kidneys, interestingly, is mostly sympathetic, which isn't to say that they don't receive parasympathetic innervation as well. They do. It's just there's an asymmetry here where many visceral organs receive dual innervation from both sides of the autonomic nervous system in about equal parts, and the kidneys don't. And this is because of the blood component. So as you now know, regulation of arterial diameter and vessel diameter is under sympathetic control only. And because the kidneys are dealing with so much blood and blood-related tasks, their innervation is likewise mostly sympathetic. So this both functions to adjust blood flow into a little ball of capillaries called the glomerulus, more on that later, but also stimulate renin secretion. So we talked a lot about the renin-angiotensin-aldosterone system and its role in correcting falling blood pressure and blood volume. And that's a system that needs to be activated very, very quickly when it's necessary. So sympathetic takes care of that as well. Okay, so I mentioned the glomerulus, and I'll talk about that in the next slide. But for right now, I want to talk specifically about the nephron. So in 241, in the very, very early parts of the lab, the first lab unit we do is to teach you the tissues of the body. So you start out by learning to distinguish between and among the epithelial types and connective tissues and whatnot. And there is one slide in particular that is used for both simple squamous epithelium and for simple cuboidal, and that is the kidney slide. So the kidney has both, because most of what your kidney is made of, the parenchyma of your kidney, which is the cells that actually perform its function, are just wadded up tubes of simple cuboidal and squamous epithelium. And the nephrons are what those tubes are made out of. So when you slice the kidney, you're just getting various sectional views of many, many, many nephrons. So a nephron is a tube into which a specialized capillary bed is plugged. The capillary bed here is called the glomerulus. I'll show you a closer picture of it in a moment. But basically, it's pressurized such that fluid is always leaking out of it via hydrostatic pressure. So not unlike the regular capillary bed that we talked about during capillary filtration. And then the rest of the tubes in the nephron are there to collect that fluid and modify it, do stuff to it. So you have about 1 million nephrons in an adult human kidney. And adult human kidneys are pretty small. So look at your fist, make a fist and look at it. They're about that big, and in some cases a little bit smaller. So I've seen kidneys in elderly cadavers that are smaller than that, in fact. So they receive 25% or so of your cardiac output. They filter your blood and make urine many times per day, and yet each individual one is no bigger than your fist. So they are tiny compared with other organs, but man, do they do important work. So we should appreciate them. So the nephrons are actually doing that work, and I will walk you through the process of creating filtrate and the process of modifying that filtrate to produce urine, because we don't actually call it urine until it gets here into the collecting duct. When it's in these tubes, we still call it filtrate because it's not quite finished yet. So of the nephrons, there are two varieties. So if you look over here, we've got cortical nephrons, and these are primarily in the cortex. And juxtamedullary nephrons. These ones have a glomerulus that sits closer to the dividing line between the cortex and the medulla, and their nephron loop, or loop of Henle, extends much deeper into the renal medulla. We will talk about the significance of this difference in more detail later, but for now, most of your nephrons are cortical nephrons, so they sit up here. And you'll see this as well in the histology video that I'll make about this same content. And then only about 15% or so are juxtamedullary. One of the reasons for this is spatial. You can only fit so many nephrons down here at this border. There's a lot more space for them to be sort of intermingled up here. So let's discuss the glomerulus. The glomerulus is a specialized capillary bed, as I mentioned, and notice the afferent and efferent arterioles are both arterioles, meaning that this is not a capillary bed at which gas exchange takes place. So it's about 50 or so capillaries, and these are fenestrated capillaries. Remember, fenestrated means windowed. These contain large openings. And they contain large openings because they're under constant pressure. So typically the diameter of the afferent arteriole is bigger, wider, than the diameter of the efferent. So this means that this capillary bed is under constant increased hydrostatic pressure. So plasma is going to be leaking out. So the function of this is to filter the blood. So large things like albumins, large plasma proteins, fibrinogen, cells, large things should be staying in. Small things and fluids, so plasma, dissolved ions, tiny dissolved nutrients, etc., those are going to leak out into this capsular space and form the filtrate, which enters a series of tubes that modify the filtrate to create urine. So filtrate is very similar to blood plasma, but it doesn't have as much protein. It should have very little. And it doesn't have any cells in it, or it shouldn't anyway, unless you've got some very bad problems like glomerulonephritis. So we'll talk a little bit about some kidney pathologies in a bit. So blood hydrostatic pressure in the capillaries is what forces fluid out of the plasma and into Bowman's capsule. So I'm going to ask you a Socratic question here, and I want you to take a moment to think about it and try to answer it. Chat's been quiet this morning, probably because you guys just woke up and are tired. Well, maybe not just woke up, but it's morning. Let's say that. Based on what you now know about capillary exchange and blood pressure, what effect do you think chronic high blood pressure has on the kidneys, specifically on this structure? So no right or wrong answers. I'm just asking you to speculate. So take a moment to think about that and then tell me what you think. Thanks for weighing in. Yes, more solutes would get filtered out. So if you think about this structure and regulation of it, It does indeed affect the diameter of the afferent arteriole. Very good. So there are micro adjustments made to the diameter of the afferent in response to blood pressure changes to try and keep glomerular filtration constant. But if, imagine a person that has unregulated hypertension. At some point, their ability to regulate their glomerular hydrostatic pressure is going to fail because global pressure in the blood is just too high to do that. What that ends up doing is putting a tremendous strain on the kidneys because filtrate is being produced and forced through the renal tubule faster than the kidneys can reasonably keep up with. So one very real consequence of chronic high blood pressure, especially if it's paired with diabetes, is a loss of renal function culminating in renal failure. So I've been mentioning sort of lifestyle-based public health issues that are specific to the United States and other developed countries, where on average our citizens have pretty good access to lots of calories, nutrition that is calorie dense but relatively nutrient poor, and other habits like over drinking and smoking, etc. And so you often see these sort of clusters of symptoms where, due to behavioral choices that a patient is making, and to some extent genetics as well, you tend to see pathologies happen in groups where somebody is going to have diabetes and high blood pressure and maybe some amount of cardiomyopathy and also blood pressure issues that are affecting their kidneys and causing renal failure. And these problems cost our country billions and billions of dollars a year. So I'm emphasizing them not because I'm, passing judgment on people's lifestyle choices, like I like wine, whatever. I also like French fries, but rather because they're public health issues that my students as a population are likely to encounter both in their personal lives with relatives and also in a clinical setting. So the more informed you can be about how all this stuff is interconnected, the better citizens you'll end up being in general. So both of you that answered are right, and thank you so much for weighing in on that. I appreciate your contribution a lot. Okay, so the last person to comment mentioned diameter of the afferent arteriole, and that actually provides me with an excellent segue to the next topic, so thank you, that person. I also want to open this with a vocabulary warning. This is the second juxta thing that you're seeing. So the first was juxtamedullary nephron. Now we're moving on to juxtaglomerular complex. So juxta just means at the boundary of, so juxtamedullary nephrons are at the boundary between the cortex and the medulla, and juxtaglomerular complex is at the boundary between the tubular area and the glomerulus over here. The JGC, juxtaglomerular complex, is an apparatus that monitors what's passing through the glomerulus and the tubules, and then makes adjustments to the diameter of the afferent arteriole. So it's a feedback mechanism that controls or tries to control the hydrostatic pressure in the glomerulus here. So there's two parts to it. One is the macula densa. So if you look at the glomerulus, let me grab my magnifying glass here. I'm also going to grab my Epic pen. Maybe. Okay. So with regard to the anatomy of the glomerulus and the Bowman's capsule, so the renal corpuscle, which all of this stuff is, and this is a short anatomy lesson, which will help you on Practical 3. There's two poles to this thing. So the whole entire structure, including the glomerulus and the capsule, has a name, and that name is the renal corpuscle. And then within the renal corpuscle, there are two poles. So this one, where the filtrate exits, we call the urinary pole. And this end over here, where regulation of flow into the glomerulus occurs, we call the vascular pole. Now, the degree to which you can distinguish between the urinary and vascular pole on a given renal corpuscle in the microscope slides very much depends on getting a lucky slice that shows both, but you can sometimes see it. It's pretty cool when you can. So the vascular pole is the end of the renal corpuscle at which flow regulation into the glomerulus occurs. The urinary pole is affected by the vascular pole. So the extent to which the urinary pole is receiving good flow of filtrate or not is affected by what the juxtaglomerular complex is doing at the vascular pole. Okay, so let's back out of my magnified section here and back to, move this over there, business as usual. Okay, so over here at the vascular pole, there is a renal tubule, and this is the distal convoluted tubule. So the filtrate that's in this tube has already passed through the nephron loop, so it's well on its way to becoming urine. In fact, it almost is. So the cells here, and if you look, we have like a cuboidal epithelium that's pretty normal and regular, and then on the side of this tube that is kind of stuck in the crook of the afferent and efferent arterioles, suddenly the cells become tall and skinny and sort of mashed together. That's called the macula densa. Macula means spot in Latin, and densa means dense. So it's just named because it's a spot where the cells are extra densely packed together. These have special sensors that detect the amount of sodium in the filtrate, and the sodium amount is used as a proxy for flow. So here's how this works. It requires a couple logical leaps, but you guys are more than smart enough to get them, so just bear with me and I'll help you through. The more filtrate is coming out, sodium's going to come with it. And the more filtrate is coming out at higher hydrostatic pressures, the faster filtrate will be formed, and the faster that filtrate will move through the proximal convoluted tubule. So bulk fluid flow increases with increased filtration pressure. So filtrate's moving faster through these tubes if this pressure is higher. If the filtrate's moving pretty fast, a lot of the pumps for sodium in the proximal convoluted tubule are not going to have a chance to grab the sodium as it passes because the sodium dissolved in the filtrate is going to be moving too quickly. So what that means is more sodium will end up in the distal convoluted tubule. So more sodium in the DCT indirectly means that filtrate's being formed really fast, which indirectly means that pressure in the glomerulus is high. So it's like if then, sort of like a Boolean logic gate sort of thing. Sorry, I turned into my dad for a second there. He's a computer programmer. That was something that he would have said. So apologies from my father, Jim Howard, for making me this way. So the juxtaglomerular cells are next to the macula densa. So basically the macula densa is constantly sort of telling the JG cells how much sodium they're encountering. And then the juxtaglomerular cells, these are actually modified smooth muscle cells. So they'll kind of tighten up or loosen around the afferent arteriole according to the signals they're receiving from the macula densa. It's also an endocrine structure. So we know that the juxtaglomerular cells secrete renin as well, part of the renin-angiotensin-aldosterone system. And we know that erythropoietin comes from the kidneys, but not specifically where. So since they secrete renin, it would make sense for EPO to also come from here. And also this is an area that senses blood flow, but nobody actually knows for sure. So there's a question in the chat, and that is, are macula densa chemoreceptors? And the answer is yes. In addition to being cells that just make up the wall of the DCT, they contain sensors that detect sodium, among other things. Absolutely. You're welcome. So quick programming note before we move on here. In the past, students have asked, well, how do you know that renin comes from here but not erythropoietin? And the answer is staining for stuff and also histological techniques around this. So a lot of times when scientists want to know what tissue hormones come from, they will just take some samples of various organs and grind them up and then do chemical assays for that particular hormone. So doing that to the kidneys results in you see calcitriol, you see renin, and you see erythropoietin. And then histological studies where you take slices of the kidney that preserve its cytoarchitecture, so you don't grind it up, and then stain for things will give you renin concentrated here. But nobody's managed to locate specifically where EPO comes from. So we know that if you grind up a sample of kidney, you'll see EPO in there, coming from there, but as far as what specific part of the kidney, not sure. Okay, so we're getting into renal physiology now. And as you might imagine, there's considerable consequence for renal physiology. So one of the things that it does is maintains homeostasis of blood volume and blood composition. So I mentioned blood volume, so you can control how much or how little you're peeing out, and that's going to have a direct impact on the volume of blood. Also composition of blood, so to what extent are ions preserved or excreted, so sodium and potassium being kind of major ions that are controlled by the kidneys, as well as protons, and excretion of wastes. Urea is most abundant, and that's from amino acid catabolism, so a lot of the stuff that we just talked about at the end of the digestive system. Creatinine, which is the byproduct of creatine phosphate metabolism. So here's a link between the kidneys and the skeletal muscle. So if you think way back to discussions of muscle, we did talk about the order of fuel use in muscle and where the muscle ATP comes from for skeletal muscle. And remember the phosphagen system, which is creatine phosphate. Creatine phosphate is a phosphate donor, so you can take phosphate off of creatine phosphate, add it to an ADP and get ATP, and then creatine results, and that's changed into creatinine and you pee it out. Excuse me, and also uric acid from RNA recycling. So you get uric acid mostly from nucleic acid catabolism. For us, waste excretion requires water. So some animals are able to pass nitrogenous waste containing urea, ammonia, and uric acid in the form of kind of a chalky paste, which minimizes water loss. Birds, for example, do this. That's why bird poop looks the way it does. When you see bird poop on your car with like the little black part and the white part, what you're really seeing is poop and pee at the same time. The white part is the bird's pee. We, unfortunately, have to lose a considerable amount of water when we remove wastes. So we have an unavoidable water loss, which means that we have to constantly be drinking water. Speaking of which, I've been chugging coffee all morning, so I thought I should take a sip of water. But also that there's a robust apparatus in the kidney that exists to oppose water loss, and there's considerable hormonal regulation of water loss as well. Sorry, this is bothering me. There we go. Processes of urine formation. So we'll focus on filtration first, and that is the process that happens at the glomerulus, as indicated by the green arrow here. After that, we have tubular reabsorption and secretion. So secretion is putting waste products into the filtrate, and reabsorption is taking stuff that is necessary to keep out of the filtrate. So we'll talk about all of these, and what we're going to kind of do is move along the glomerulus while we talk about it. So let's discuss filtration first. This portion of the physiology of the kidney has a lot in common with the capillary filtration that we just talked about. So we're going to have to do things like worry about hydrostatic pressure and colloid osmotic pressure, respectively. So hopefully your investigations of capillary filtration have prepared you a little bit more for thinking about those topics here. So we know that the glomerulus is under pressure. One thing we haven't talked about yet are what these weird little squiggly guys are. These are called podocytes. So the capillaries of the glomerulus are fenestrated, meaning windowed, and then they're covered over with cells, and these cells have feet that wrap around each other and interlock to form filtration slits. So these little feet are called pedicels. So they basically help to control what is able to filter out into the filtrate and what is kept inside of the glomerulus. So healthy podocytes, podocytes are also called the visceral layer of the Bowman's capsule, with the outside part being the parietal layer. So plasma and little solutes, so small ions and such, are forced into the capsular space out here because they're little enough to squeeze through these filtration slits. So this is the result of two pressures: hydrostatic pressure and osmotic pressure. So let's break those pressures down in the next slide. So due to the interplay between blood pressure normally and the juxtaglomerular apparatus regulating the diameter of the afferent arteriole, we've got glomerular hydrostatic pressure staying at about 50 millimeters of mercury. The capsular hydrostatic pressure, because of course there is fluid in here, it's not full of air and it's not collapsed, there's always some amount of fluid in the capsule, so that fluid is exerting pressure as well, is only about 15. So if you were to ignore colloid osmotic pressure, you would get net hydrostatic pressure alone of 35 millimeters of mercury. But we can't ignore osmotic pressure, so we have to look at that as well. So the blood colloid osmotic pressure, assuming, normal, is 25 millimeters of mercury. The filtrate colloid osmotic pressure, by comparison, is 0. So although there are solutes in it, they're not in an amount that's appreciable compared with the high solute activity in the blood. So 25 versus 0. So once again, to calculate net filtration pressure, in total, you take net hydrostatic, which is 35, 50 minus 15, minus blood colloid osmotic pressure, which really should be net colloid osmotic pressure, but because filtrate colloid osmotic pressure is considered to be 0, the 0 falls away and you just have that 25 by itself. So this means a net filtration pressure on average of 10 millimeters of mercury. So that's what keeps fluid driving out of your glomerulus and into the urinary pole and on. This brings us to an important clinical measurement, and that is glomerular filtration rate. So this isn't talking about how much urine you're producing, but rather how much filtrate. So regardless of how much or how little you are urinating out, your GFR should remain roughly steady. And that's because you need to constantly be filtering wastes out of your blood. So we talk about this as the amount of filtrate produced per minute, and this is about 125 milliliters per minute. So about 10% of the volume of blood that arrives at a kidney ends up as filtrate. So if you multiply this times the minutes in an hour, you get about 125 mils per minute times 60 minutes in an hour. The minutes fall away via reciprocal crossout, and then you get 7.5 liters per hour. Now, are you peeing 7.5 liters per hour? God, I hope not. You would dehydrate to death, but that's how much filtering you're doing. If you then multiply that times how many hours there are in a day, you're filtering your blood in the amount of about 180 liters per day. So think about cardiac cycles, think about blood moving into the systemic circuit, and 25% of cardiac output. Obviously, you don't have 180 liters of blood, right? So what that means is you are sending the entire volume of your blood through your kidneys multiple times per day. You're constantly cleaning your blood of the wastes associated with being metabolically active. So 180 liters per day works out to be about 70 times the amount of total plasma volume. So this is considerable. And if you're just looking at these numbers here, imagine what happens if the kidneys begin to fail. This should show you just how quickly dangerous amounts of waste products are going to build up in the blood and really drive home the need for example, hemodialysis in the cases where somebody's kidneys aren't functioning and they haven't gotten a transplant yet. So there are people in our population here in Portland that have to go get hemodialysis every single day. So for those of you who are not familiar, hemodialysis is where a machine takes the place of your kidney. So you go to the dialysis clinic, you lay in a bed, you have your blood hooked up to this machine, and your blood goes through the machine and it's cleaned basically and returned to your system. So for people that need daily hemodialysis, folks that have zero renal function, they basically spend all day, every day in the dialysis clinic. So there's a really profound consequence on quality of life for those patients. And, you know, for my students who end up being nurses, it's possible that you may end up working in a hemodialysis clinic and monitoring the health and well-being of the dialysis patients. So it's something to be aware of for sure. And also, this is just an anecdote before we take our little break, but I have noticed that the number of dialysis centers that have begun popping up in Portland as new development has increased considerably over the past 10 years. I keep seeing like DaVita kidney centers like suddenly appearing all over the place, which I'm guessing is related to the demographic increase in age among our population. So we're gradually, as a population, getting older. So it's probably related to that. But it's a very big industry, which means that it's a place where jobs are available. So, all right, so I'm going to pause here and take another short break. And when we come back, we will continue discussing how glomerular filtration is regulated.